Hey everyone, it's great to see people from all over the world joining in today. I think it's time to get started. Good morning, good afternoon, good evening, and good night for some of you. And welcome to today's webinar, FixNCut, Reversible Tissue Fixation and Dissociation for Single-Cell Assays, here at our webinar series at Bioprotocol, which is an open access online journal with the mission to make research more transparent and reproducible. Just a few comments so you get the most out of this webinar. If you have questions, you can ask them or post them in the Q&A or in the chat section throughout the talk, and we will address them at the end. And this webinar will also be recorded, and so you can revisit it later on our website. Okay, it is my distinct pleasure to introduce today's speaker. We have two, Laura Jimenez Gracia and Xiu Wang. I hope I pronounced that right, or close to right. Laura is a bioinformatics researcher who currently works as a postdoctoral researcher at the National Center for Genomics Analysis in Barcelona. She earned her PhD in biomedicine from the University of Barcelona, focusing on human inflammation and immunity at single-cell resolution, contributing to advancements in translational research. She specializes in single-cell sequencing data analysis in the context of immune-related diseases and cancer, and her expertise spans single-cell transcriptomics, immune profiling, and other omics. Xiu is a senior scientist in the Spatial Technologies Unit of the Precision RNA Medicine, or PRM, core at the Beth Israel Deaconess Medical Center. His scientific training started in Marburg in Germany, and he completed advanced training in cell and developmental biology at the University of Osnabrück in Germany, in Basel in Switzerland, and at the Weizmann Institute for Science in Israel. Before joining the PRM core, he held a staff scientist role at the NYU School of Medicine and Harvard University. And his research focuses on advancing spatial transcriptomic technologies, understanding pathology in genitourinary oncology, and investigating mechanotransduction in development and diseases. And with that, I would like to pass it on to you two.
Thank you.
Thanks a lot for this nice introduction and also for the opportunity to present our work here. I will start the presentation and we will switch when we present more like the wet lab part or the computational part. So let me start presenting the two papers that are related. One is the methodology and assessment. That's the one that was published on Genome Biology, and the one that brings us here is the protocol that was published in Bioprotocols. But everything started in the context of single-cell data, because sometimes we realize that when we are working on decentralized studies, that we need to disconnect time and location for the downstream processing steps. Some samples were suffering. We were not able to recover proper data. Then we also know that the cells the transcriptomic profile of cells is not constant. It varies with different external factors that can stress what's the real state of the cell. And then, even though the cryopreservation is like the gold standard in the field and it preserves the transcriptional profile of cells, we realize that sometimes in solid tissue, the composition of the cells might vary, and some cells that are more sensitive to external stress might be lost during the sample processing. So in this context, we aim to establish a standardized protocol for sample collection, cell stabilization, and tissue processing that was compatible with single-cell genomics technologies. And that's why we are here now presenting the FixNCut workflow, which uses the Lomand reagent, or also called DSP, that will enable the reversible tissue fixation and sample dissociation. And basically, those are the steps that we will be following. That's we collect the sample, then we fixate this sample with the DSP. We can, if we need long storage, to cryopreserve for like either transport or either like preserving for a long term. Then we will perform dissociation, and then we can already work on the single cell genomic assay. But I'll hand over Shushu, who will tell more you about how to like really implement this protocol.
Yeah, so thank you, Laura. So in this protocol, we have included the technical details in the manuscript, but I'm just going over the hands-on part. So basically, this protocol utilizes the DSP to cross-link the amines containing amines from proteins. So this is the similar processing principle as you are familiar with the formaldehyde fixation. So it generates amine bonds and stabilize the molecules. And then, but the DSP is NSH is used in this protocol it's rather like volatile. So the most challenging part is the preparation of the so-called working solution that I'm going to show with the video in the next slide. But here in this slide, you show this is practically everything what you need. You need DMSO to dilute your DSP and make the working solution that you can freeze and store. And then in the middle, what you see is like basically tris buffer, because tris has a free amine to neutralize the DSP, because you need to do that before you store. And then you have the other things what you need, basically a buffer and or PBS concentrate or just normal PBS. And then next, and this is we call it a version 1. And next slide, please. So basically on the left side, there should be does it work? Yes, okay. So on the left side, there's a little movie showing how to prepare this concentrate, this work solution, how to use this work solution. You need a vortex, and you just turn it on, and you have your working solution that you can see that's in the bottom of the tube, and you add job by job, which is very critical step, that you add the PBS, 490 microliter PBS job by job into the 10 microliter of the stock solution. Here you see that. You really literally add job by job. This is because the DSP has a very limited solubility in water. If you add too fast, it will precipitate, and you may see some crystals. So this is something we can discuss at the end. So the most important part is to add slowly and really count the jobs, and then at the end you look at the tube, see whether it looks clear or you have precipitation. So if you have precipitation, you may need to repeat the whole process. So this is what we show is the working solution, and then the fixation is done by adding this work solution, the fresh work solution, to the cell suspension, and following the protocol that we have in the publication. It takes like 30 minutes. It takes 30 minutes usually for the cell suspension. For tissue, it's slightly different. You may need to optimize a little bit, but it's important to mince or cut the tissue into small pieces so that the DSP can penetrate, and to ensure a good fixation step. So and then the last step, what you need to do is to add the tris buffer to neutralize. And once it's neutralized, you can store it and freeze it as you are used to, and in a -80 freezer. And then you have, because there are two options, you can store them or you can immediately process them on different platforms, not limited to the Chromium X that is in the figure. Next slide, please. So recently we have posted the protocol we call FixNCut protocol version 2. But it's important to emphasize that version 2 does not mean it's a better version or advanced version of the version 1. It's rather a different protocol. So before you try that, you have to see what fits your needs better. Because as I mentioned previously, the DSP NHS ester has a solubility issue, which means, for example, now if you have 8 samples that you're collecting at the same time, would be a little bit cumbersome to prepare the working solution, because you cannot prepare a large master mix, because otherwise it will have a precipitation issue. So then in this protocol, we use a different strategy. We use the methanol, which can dissolve the DSP much better and stabilize it. Because in the methanol, the NHS ester is less prone to degradation, which also has a complementary function of stabilizing cells, because probably you have heard that, or you have used the methanol, cold methanol itself as a fixative. So the strategy here is to combine the cross-linking capability of the DSP with the intrinsic like fixation function of methanol and utilize it as a stabilizer. So this combination will provide a good improve the cell integrity, minimizes clumping of methanol itself, and makes the processing much simpler, and allowing you to preparing a larger master mix for more cells at once. We have one of the benefit or potential benefit of this is that you can also process, for example, like a contiguous or like samples such as, like, for example, COVID samples, influenza samples, HIV samples, that are usually less easy to preserve and to store. So in this slide, I also attached the QR code on this. I also have the QR code, so you can scan if you want, and you can check the updated version. And also there's an image of Dr. Matalotto demonstrating the single cell techniques. And he is actually the person who developed this protocol, and we are very happy to adapt this protocol very early. And we'll be happy to take hands-on questions at the end if you have any questions regarding how to adapt this protocol to your own sample and your question. Thank you.
And now, after you already know the stars of the movies, I will try to keep your attention. So I will explain a bit how we validated that FixNCut indeed worked. So basically, and that it was compatible with single cell analysis, and of course that it preserves the natural state and also reduce the technical artifacts that you might generate during sample processing. So first of all, we start by validating it with human blood samples, and here assessing 2 conditions, fresh and fixed conditions. And what we observed is that we were able to recover same amount of exonic sequencing reads, and at the same time, the number of genes that we were able to capture at different sequencing depth were comparable. So FixNCut preserves the ability to detect all the gene expression profile that we expect from single cell transcriptomic data. At the same time, we were able to recover all the immune cell profiles from blood. So it allows us for a full phenotyping of the cells. And here we did not perform sample integration. And as you can see, we do cluster cells by their identity and not by the protocol that we apply, fresh and fixed. And in terms of cell composition, we were only assessing 2 samples, so no replicates, and only couple of immune cell populations show slightly changes. The other ones were fully comparable. In terms, digging a bit more in terms of the gene expression, we perform a pseudo-bulk gene expression where each dot here is a gene, and the correlation that we found between the fixed and the fresh sample was almost near to 1. And when we assessed for differential gene expression between the 2 conditions, we only found 4 differential expressions that were associated to hemoglobin genes and mitochondrial. That was a phenomenon that was also been described before when performing digestions at low temperatures. Furthermore, in order to assess the fact that FixNCut can reduce the cell stress artifacts, here in the ThenAC single cell genomics group, we have previously defined a sampling time related signature assessed at different times where samples were cryopreserved. And based on this sample, we try to see how our differential gene expressions upregulated in the fresh condition were overlapping with those sampling time gene signature. And as you can see, there were almost like 30% of the genes of this signature were captured to be upregulated on the fresh one compared to the fixed. And if we see how this signature applies to the different immune cell populations that we have defined, in general, we do see how all the T cells from blood are the ones that are particularly affected. So this DSP is protecting the most those T lymphocyte cells. Moving on, as we say that it's a beneficial for especially human and mice biopsies, we wanted to see how DSP and FixNCut workflow perform in solid tissue samples that needed to be digested. So for that, what we assess is two different tissues, lung and colon from mice. And again, you can see how lung cancer allow us to recover a comparable number of sequencing reads, and as well the number of detected genes was similar. And in the case of colon, that it's even a stronger a tissue that it's known to be more affected by the stress, especially on the epithelial and stromal cell fractions. You can see how the fixation indeed allow us to recover many more reads that map to the exonic regions of the genome, and also the number of detected genes that we recover was larger. Moving into the capacity to identify and define different cell types, again, we see that we were able to recover all the cell types that were expected, both in the immune fraction and the non-immune fraction. But as you can see here, the profile was so different that an integration method between the two protocols was needed in order to define common cell phenotypes. And here it's interesting to see how the fixed cell shows a different profile on the fresh sample. Especially, we do see an expansion of epithelial and stromal cells compared to the fresh one. And some of you might think, why is the fixed the one that we trust the most? Is because if we compare that with experiments in which we assess single nuclei transcriptomics, that's the way to overcome problems on sensitive tissue samples. These ones are the ones that tell us that the largest stromal fraction the largest percentage of stromal and epithelial fractions is the one that we should expect from these samples. And again, here we wanted to assess in sample biopsies, we also observed this reduced stress response. And for that, we use a dissociation signature that it's linked to dissociation using worm collagenase. And here I'm showing the results across all the cell populations that we define both in the lung and in the colon sample. And as you can see, there is the fixed samples, especially on those related to stromal and epithelial cells, show a strong, significant reduction of the cellular stress of those samples. And of course, the more sensitive the tissue, the more reduced stressful conditions and the better capturing all the populations we are expecting to see this effect. Then, until now, we were only assessing fresh and fixed samples. But at this moment, what we try to assess if that FixNCut protocol was also compatible with long-term tissue sample storage, meaning for like ages as normal samples can be stored. And for that, we added an additional condition, that was the cryopreservation after fixation, that was assessed on lung mice tissue. And as you can see here, for instance, on the pseudobulk gene expression, we do recover almost a perfect correlation on the gene expression between all the three conditions. And again, in terms of gene cell phenotyping, we do recover all the cell types that were previously described and capture only assessing fresh and fixed samples. Moving a bit on into like the multi-center clinical research study, what we did is to move from this mice tissue assessment into a real human biopsy assessment. And for that, we use colon biopsies from patients with IBD in remission. So we added this additional layer of working on a hospital. Here again, we assess all the conditions already presented, so fresh, free, fixed, cryopreserved, and fixed, followed by cryopreservation. And as you can see, the number of genes recovered is comparable across all the 4 conditions. The correlation of the pseudobulk gene expression is very high across all conditions. And in terms of the gene signature score associated to the stress-related signals, we do see how fixed reduces compared to fresh, and the fixation followed by cryopreservation reduces the signature compared to the gold standard cryopreservation way of preserving samples. To move a bit further and generalize the applicability of FixNCut, we assess if that was applicable with other single-cell assays in which we involve cell labeling, either with antibodies, either with lipid targeting the cell membrane. So, for instance, to multiplex multiple samples into the same library preparation system. So for that, what we assess is we did a FACS sorting of cells labeled with different antibodies. And as you can see here, for the CD45, that's capturing the immune fraction, CD3 that will tag T cells, and CD4 and CD8 that tag the 2 different T cell subtypes. We do see that we are able to recover a good signal for both fresh and fixed conditions. Here I'm showing the mean fluorescence intensity result of the FACS sorting, and the same was done assessing for lipid modified oligos. That's a way that 10x Genomic uses to pull different samples into a single gem on a chip. And here again, you can see the comparable of the signal between fresh and fixed. So basically, even though you see that there are some reduction on the signal on the fixed, that's not a problem, because what we are suggesting is that using the same amount of antibodies, we are able to capture both signals. But of course, different protocols might need some adjustments on the amount of antibody that you are providing. And last but not least, we also wanted to see how this was applicable to spatial omics experiments. And here we're showing a prostate cancer biopsy that has been imaging with a PhenoCycler using from Akoya Biosciences. And we are comparing the gold standard formaldehyde paraffin embedded with the DSP paraffin embedded counterpart for both the hematoxylin and eosin staining, and several markers associated to the tissue structures. And as you can see, the results are pretty good in both of them. So with that, I will just go into the conclusions. So the FixNCut protocol combines this reversible sample fixation with subsequent tissue dissociation. It preserves the cell transcriptome, minimizing the stress-related technical artifacts that can arise during the sample processing, ensuring that we have a good RNA integrity, that the library complexity is equal or even better, to preserve the real tissue cellular composition and the gene expression. Then we enable this time removal and location constraints from the sampling to the processing sites. And it also allows staining with antibodies, lipid-modified oligos, enabling all the variants that single-cell technologies offer us, not only cell labeling and hashing, but we also predict that it will work with good results with site-seq or other chemistries, and that it can also be applied in the context of spatial proteomics and transcriptomics. And with that, just say that FixNCut it's offering versatility in the field of single-cell technologies applications, and will definitely help to design robust and flexible studies in the context of clinical research. And that's not part of our work, but those are some work that has already been published, working on pancreas tissue that's well known to be very sensitive tissue, and until FixNCut was released, was also always explored using single nuclei, single cell transcriptomics. And these are some tissues and samples that we are aware that are being currently processed with FixNCut workflow, but still are unpublished work. So it has been successful for brain metastasis on human biopsies, adult brain metastasis from mice models, to characterize organisms from the Cnidaria phylum, to process urine cells from humans, in liver biopsies from rat and mouse, and also working on sorted T cells in human and mouse samples. And with that, thanking not only Shushu for presenting together with me, but of course all the team from Broad Institute, from Australia, together with Luciano Martellotto, and from the Single Cell Genomics Group here in Barcelona, that allow us to do this study and to assess across multiple tissues and organisms. And we will be more than happy to take any questions now.
That was really fascinating work. Okay, we had a few questions in the Q&A chat section, but I'm gonna wait. Okay, actually, let me go. So Rishit Panchal asked, great presentation. My question is, given the FixNCut relies on reversible cross-linking for sample preservation, how does the protocol ensure complete reversal of fixation without compromising the RNA and DNA integrity? And there's multiple questions, are there any risks of incomplete reversal that could lead to lower sequencing efficiency, data loss, or biases in gene expression? And additionally, what quality control measures are in place to verify successful reversal across different tissue types and single-cell assays?
Or do you want to take that question, or?
Go on it.
So basically, we wrote in the manuscript that's also posted in the chat just a couple of minutes ago. So the reversal it's not essential, or in many cases it's not needed, right? So if you are going to run single cell transcriptomics or spatial, they can be there in place, because for spatial it's very obvious, because it's you need the tissue to be intact, and because the cross-linking is only introduced between for example, the lysines in your amino acid chains, it doesn't interfere with the DNA or RNA, right? You can revert it with DTT. For example, when you're running the single cell, you already add the DTT in some, for example, in a 10x kit, it's called a reducing agent. It's the same thing. You're already reverse that and lysing your cell, so you don't really care about the cross-linking. So it can be done, but as we showed, but also other authors showed, because it takes more time, because you need to incubate in your wash, and you may lose your samples or lose your cells. So you have to evaluate if that's necessary for your downstream analysis or not. Right? I hope that answers the question.
Rashini said, very nice talk. I wanted to know if the imaging data was quantified for fluorescence intensity, and as one would expect, using DSP would make antibody permeability more complex. Was that the case?
I think I missed the first part of the question, but it was related to the FACS sorting, right? Of the using antibodies when the sample was fixed.
Maybe, are you still there, Rachind? Maybe you could add on to that.
Or maybe was that talking about the spatial, the imaging data? So
All right, let's pass on to the
Okay.
next question. I'm sorry. Any comments to adaptability to plant samples, for instance, fixed tissue and then protoplasting for single cell sequencing? Are you aware of anyone having tried that, or do you foresee complications on some of those steps?
We haven't tried, but I can imagine how that can be useful, especially for plants or for fungi. You protoplast in them and you go through the same protocol. It should work. Yeah.
Someone said, hello, I've tried DSP fixation in HeLa cells, but it seems that the cell prefers to aggregate and be lost. Have you met this problem before?
We tried that. We don't see aggregates. I think Laura's lab also tried HeLa's or other culture cells. No?
I think it was Luciano who tried. We haven't tried them here. But losing cells on aggregates, I haven't experienced, but sometimes what happens is that if you observe the cells under the microscope, the fixation might make you think that cells are dead, and you might not consider those cells. But indeed, that's part of the fixation that cells look like that, but they are not really dead. So you should not discard those cells.
Yeah, because the thing is, aggregates for cell aggregates, I was trying to check how, for example, like viability before you do the fixation, right? So and then we try different cells and we don't really see aggregate. Also, very important to check is your DSP fresh, and is the DSP work solution fresh, and then yeah, I personally happy to discuss about that in later or happy to troubleshoot. Yeah, but we don't really see that.
Gabriel de Barros, right, asks, for spheroids of about 1,000 to 10,000, would you recommend the method of cell fixation or tissue fixation?
I think it depends on the downstream assay and depending on the logistics, because we often do the tissue fixation when we harvest either from rodents model or from surgery. So we do because very often the limitation is the logistics, because sometimes there's no chance that you can process the cell dissociation on the spot, especially in the clinic or in the animal facility. So that's why this protocol was developed. So in that case, it really depends. So if you have the infrastructure, of course, you can go to the cell fixation, but otherwise tissue fixation. So this is a logistical question. Both should work.
Thank you. Georgios Raphael, Samantidis, I'm so sorry for mispronouncing all of your names, possibly, said, thank you very much for this presentation. Could FixNCut be adopted for a smaller system, such as flies or mosquitoes?
I think so. I mean, I don't know what about Laura's team, but we haven't tried because we're like a medical center. But I personally have worked more than 10 years on the fly, on the insects, right? So I don't see why it shouldn't work if the cells are, if you can specify what type of tissue you're working on, in fly or mosquitoes, I can probably provide a little bit more information. But happy to help, happy to discuss how to adapt that protocol, but it should work.
From the internet, some people have previously asked questions. Alexandre Parola asked, is FixNCut compatible with, for example, proteomics or lipidomics analyses downstream?
I think Laura's lab probably has more.
We haven't performed any like proteomics or lipidomics studies because we are not focused. We are more genomics team, but I think should be feasible. What I'm not sure if there is a reversible step that should be performed, that it's not automatically performed, as in 10x, for instance, single cell 10x technologies, that there is this DTT during the reverse transcription, so that you don't really need to kind of automatically, like manually add some step. But I think as far as you are aware of this step, should work.
Yeah, I think it's I think we haven't tried yet. But that being said, for example, if you are doing like proteomic, yeah, it's like I think it might be the reversal might be important too, so that you get your confirmation right. I think we are for the I think this we haven't tried. We haven't tried it.
Okay, thanks. Jared asks, have you tried the kit with Parse Bioscience single-cell RNA workflow? And is the Parse fixation protocol still needed if you're using FixNCut version 2?
Yeah, so so this is I think the Parse workflow is very similar to what we have. I think the major difference that Dr. Matalotto developed is that we have this kind of more like open protocol. It's essentially the principle it's the same. So so I think what I would do is, I mean, so let's say it this way. So the FixNCut protocol can be adapted not only to 10x genomics, right? It can also work on the Parse on an alternative single cell platform. So that's people have already tried. So the principle it's the same as what Parse allow now the or the Illuminati fluent ones. So it's the same protocol essentially. We have different details about the timing, concentration, and wash neutralization. That's other variants. But I think the most important thing is to try on your own samples to see which one works the best. We may need to optimize.
We might have covered all the questions that I can see. Oh, there's one more. Have you tested how long cell and tissue are stable at room temperature for downstream single-cell RNA?
One of the tests that we performed, it was fixing cells, but not cryopreserving them. And 7 days, like if you forget the sample 7 days into the bench, we could still recover like kind of acceptable transcriptomic profile, be able to define the different cell populations that was assessed on lung and colon mice samples. So of course it was not same quality as if you cryopreserve or you like kind of fix and process just staying at 4 degrees overnight, but you still, like in the worst case scenario, will get some data from that.
Thank you. Thanks.
But I would not recommend going for this approach.
So in general, I think this protocol is more for, as we illustrated in the paper, it's more for you harvest and you freeze them and you waiting for later processing. So if you, for example, we mentioned that in the introduction. So there were situations, for example, samples are collected in Africa, but they're going to be transported to the United States. And in that case, DSP, unless you can keep the samples frozen, DSP is probably not the most stable way. So I hope that helps.
There's one question regarding solubility into the tissue. So since DSP has limited solubility, do you think using an alternative having better solubility might make this current protocol better?
Yes, so that's how we call it version 2. It's because usually the NHS esters usually have a higher solubility in the methanol. That's why we use the methanol because methanol itself can fix tissues, right? Cold methanol. So so that's the current version. They could be, I mean, it's also possible to use alternative solvents, but they are going to interfere even more with the cellular context, more than methadone. So that needs to be tested.
Yeah. Let's try this. The one question about fluorescence intensity, there was a clarification. So have the imaging data been quantified for fluorescence intensity? And would you expect that the DSP would make antibody permeability more complicated for biopsies or FACS?
For FACS, we assess on human colon samples as well. And as shown with the human blood samples, the only we saw that for some antibodies, the mean intensity that we capture using the same concentration of antibodies compared to the fresh was a bit lower, but that would also apply when working with different tissues. So you just need to adjust if you want to recover exactly the same amount, so loading a bit more antibody, but the capability of being captured, it's almost the same. For the spatial image that I show, we did not quantify, or at least I did not receive the data of quantification since the experiment was not performed in the lab.
Yes, I think I just hope I understand the question correctly. So I think in the paper itself, in the introduction, also we've also we cited one paper from the PNAS that they actually used this method not for spatial, not for single cell, but they to do intracellular like protein detection. So that works. I think also this is related to one of the other question with the gold particles. I think the most crucial thing is that after the fixation, the cells, its DSP needs to be quenched. So it's the cell is to be, you need to neutralize it properly with the trace buffer, and then it shouldn't interfere with the rest of your assays unless let's say your epitope is kind of cross-linked, right? That's probably will limit the application, but otherwise it should work.
One more question. Is there any specific or significant variables to take into consideration to approach the dissociation step after fixing cryopreservation?
I think in general, this protocol needs to be adapted onto the system. You look at because after the fixation, the cells are the cells will be permeabilized. So you cannot assess the viability, and what the viability won't be relevant. I think it's really need to be optimized on your own samples, like look at morphology, integrity, and then you have to run. Unfortunately, we probably will run some pilots experiments to make sure that it works. That's working well.
All right. I
think probably the question was asking more about like dissociation itself. This kind of dissociation, the time and how much enzyme you use, what's the temperature, and whether you do have including any like mechanical disruption. So this something you can sometimes you can adapt from established protocol, but we try a lot to optimize. So there's no general answer of that.
Thank you. Thanks. Okay, I think this might be it for questions. So, and every time I say that, there's another one popping up. How effective is that fixation for membrane proteins? And I know you two haven't focused a lot on proteins, so I'm not sure if this is a question that is easily answerable, but I'm gonna throw it out.
We haven't specifically looked a lot on the membrane proteins, because very often what we do is we even dissociate tissue and extract nuclei to run single nuclear RNA-seq. specifically for the membrane, I cannot answer this question, but maybe so for membrane protein, maybe this published version, the V1 would be more suitable because V2 has methanol that's going to permeabilize the membrane and disrupt the membrane proteins. So much for now.
All right. Thank you to both for a very interesting talk and for being here today and answering all these questions. This was really fascinating, and there's already messages coming in. Thank you, great discussion. There were quite a few messages earlier saying thank you, great